Hello students. Today our class is on the medical microbiology from the historical point of view. We are going to study today about the different scientist contributions in the field of medical microbiology. If we go date back 200 or 300 years back, the modern microbiology we see today is not what we are observing. So it was the hard work and dedication of our renowned scientists and researchers whose contributions not only led to the discovery of this new and innovative field, but also laid the foundation for the diagnosis and treatment of some of the deadly diseases. So we will see today that what are the pathway which have been seen by the today by the modern microbiology. So continuing on today's topic, we will discuss some of the important events from the history of the subject. See, basically, microbiology has been divided into different branches. As we all know, it may be a soil microbiology, agriculture microbiology, general microbiology, medical microbiology. So here, basically, I'm focusing on the medical microbiology field. And medical microbiology is that branch of medical science which is concerned with the prevention, diagnosis, and treatment of infectious disease. In addition, this field of sciences studies various clinical applications of microbes for the improvement of health. And there are four kinds of microorganisms that causes infectious diseases: bacteria, fungi, parasites, and viruses, and one type of infectious protein called prion. History of microbial diseases had undoubtedly played a major role in historical events such as the decline of the Roman Empire and the conquest of the New World in 1340. In 1347, plague or Black Death struck Europe with a brutal force. The Roman philosopher Lucretius, about 98 to 55 BC, and the physician Girolamo Fracastoro, 1478 to 1553 AD, suggested that disease was caused by invisible living creatures. So this was the first time that a vague idea about the microorganism came into existence. So at that time, there was not any concept of bacteria, fungus, or parasite, but it was observed by the scientists, or they came to the conclusion that there may be some invisible microorganism which are causing those diseases. The first landmark in observation of these microorganisms came in the year 1976 by the Dutch microscopist Antony van Leeuwenhoek, who observed that bacteria and other microorganisms using a single lens microscope of his own design. In 1796, further landmark was seen in this field when Edward Jenner developed a method using cowpox to successfully immunize a child against smallpox. This basically was the time when the microorganism was used or utilized for treating the different type of diseases. We will discuss all these in detail in our topic. So coming to the first important contribution was through the Antony van Leeuwenhoek, whose life period was 1623 to 1723, a Dutch microscopist. He was the first person to observe bacteria and protozoa. His researches on lower animals refuted the doctrine of spontaneous generations. Earlier, the theory was given that these microorganisms come from usually some items, some food decaying parts of any uh, food items or any plants, decaying plants. and they arise from them. But it was the first time that, and this was called as the doctrine of spontaneous generation. So it was the first time that this spontaneous generation came under questions, and it was felt that all these microorganisms have some life, and they are multiplying, or they are in this environment through, and they are not arising or growing through any type of, on any vegetative methods, or spontaneously they grow. They have some type of progeny, and his observations helped lay the foundation for the sciences of bacteriology and protozoology. So we will divide this the era of the different discoveries and development in different centuries. So first we will see the era of 1400 to 1700s. So from at least the 15th century, people in different parts of the world have attempted to prevent illness by intentionally exposing healthy people to smallpox, a practice known as the variolation, after a name for smallpox, that is the La Variole. Some sources suggest that these practices were taking place as early as 200 BCE. In 1721, Lady Mary Wortley Montagu brought a smallpox inoculation to Europe by asking that her two daughters be inoculated against smallpox, as she had observed practice in Turkey. In 1774, Benjamin Gesti makes a breakthrough, testing his hypothesis that infection with cowpox, a bovine virus which can spread to humans, could protect a person from smallpox. In 1796, English physician Edward Jenner expanded on this discovery and inoculated 8-year-old James Phipps with matter collected from a cowpox sore on the hands of a milkmaid. Despite suffering a local reaction and feeling unwell for several days, Phipps made a full discovery. These all discoveries led to the foundation of modern vaccination procedures. In 1796, Edward Jenner was the first scientist who established scientifically that the cowpox virus helps in the prevention of smallpox diseases. 2 months later, in July 1796, Jenner inoculates Phipps with matter from a human smallpox sore in order to test Phipps' resistance. Phipps remained in perfect health and became the first human to be vaccinated against smallpox. And thus the term vaccine was coined, taken from the Latin word for cow, that is vacca. The 1800s. See, when we classify the development of the microbiology in different centuries, the landmark for the discovery was the year of the 19th centuries. So this was the century where several renowned scientists had made immense contribution in the field of study of microorganism and the treatment from some of the deadly diseases. So we will discuss some of the important discoveries and inventions which were made during that period. In the year 1872, despite enduring a stroke and the death of two of his daughters to typhoid, Louis Pasteur creates the first laboratory-produced vaccine, the vaccine for fowl cholera in chickens. In 1885, Louis Pasteur successfully prevents rabies through post-exposure vaccination. The treatment is controversial. Pasteur has unsuccessfully attempted to use the vaccine on humans twice before, and injecting a human with a disease against agent is still a new and uncertain method. See here, the first attempt was made by Edward Jenner, and it was a successful attempt. And after that, the major contributions in the field of vaccination was the from Louis Pasteur. So he made some of the important vaccination methods in the called for the fowl cholera in chickens, as well as for the rabies through post-exposure vaccination. And one thing it is to remember that Pasteur was not a medical doctor, but despite the risk, he begins a course of 13 injections with patient Joseph Mister, each containing a stronger dose of the rabies virus. Mister survives and later becomes the caretaker of Pasteur's tomb in Paris. So basically, this the difference between the work of Edward Jenner and Louis Pasteur in the field of vaccination was that the Jenner used cowpox virus to prevention against the smallpox. So it was the pre-exposure type of vaccination. But Pasteur, while attempting the for the discovery of rabies vaccine, he did the experiment on his focus was basically on the post-exposure vaccine. Means that a person infected with the rabies and after infection, how he can be prevented. So this was done on the patient called Joseph Mister, and he gave 13 injections with increasing dose of rabies virus, and surprisingly, Mister survived the procedure after the procedure, and he also became the caretaker of Pasteur's tomb in Paris. In 1894, Dr. Anna Wessels-Williams isolated a strain of the diphtheria bacteria that is crucial in the development of an antitoxin of the disease. So here we can see this is the picture of Edward Jenner, 1749 to 1823, and here you can see the method of vaccinating the child against the smallpox diseases. So coming to the story of the Edward Jenner, here in the year 1796, a dairymaid, Sarah Names, consulted Jenner about the rash on her hand. He diagnosed cowpox rather than smallpox, and Sarah confirmed that one of her cows, a Gloucester cow called Blossom, had recently had cowpox. Edward Jenner realized that this was his opportunity to test the protective principles of cowpox by giving it to someone who had not yet suffered smallpox. He chose James Phipps, the 8-year-old son of gardener. On 14th May, he made a few scratches on one of James' arms and rubbed into them some material from one of the pocks on Sarah's hand. A few days later, James became mildly ill with cowpox but was well again a week later. So Jenner knew that cowpox could pass from person to person as well as from cow to person. The next step was to test whether the cowpox would now protect James from smallpox. On 1st July, Jenner vaccinated the boy. As Jenner anticipated and undoubtedly to his great relief, James did not develop smallpox, either on this occasion or on the May subsequent ones when his immunity was tested again. The technique of introducing material under the skin to produce protection against disease became universally known as vaccination. As I already told, it was a Latin word derived from the cow in Jenner's honor. He quickly developed techniques for making matter from human cowpox pucks and drying it onto threads of glass so that it could be widely transported. Next, the important discovery was from the Joseph, and again then in 1857 Louis Pasteur also designed vaccine against several such as anthrax, fall, cholera and rabies, as well as pasteurization for food preservation. Next important discovery was made in this field was from Joseph Lister in the year who lived around 1827 to 1912, who is also called as the father of modern surgery or antiseptic surgery. So he discovered that most of the infections is transmitted with by the process of instruments we use and contaminated instruments, basically the contaminated instruments we use during the process of surgery. So what he did that he sterilized the instruments with diluted carbolic acid, and using it to clean wounds post-operative infections were reduced, making surgery safer for patients. The history dates back that in 1864, while working at Glasgow University as professor of surgery, Lister was introduced to Pasteur's germ theory of disease, and he decided to apply it to the problem of surgical infections. So, antisepsis is the method of using chemicals called antiseptics to destroy the germs that cause infections. Joseph Lister found a way to prevent infection in wounds during and after surgery. He was the first to apply the science of germ theory to surgery. We will discuss in detail about the also about the what is germ theory, which was given by Louis Pasteur later. Lister's antisepsis system is the basis of modern infection control. His principle made surgery safe and continue to save countless lives. He looked for ways to prevent germs from entering a wound by creating a chemical barrier, which he called an antiseptic, between the surgical wound and the surroundings. The chemical he chose to use was carbolic acid, which killed the germs on contact. So, coming to the important part or important contributions of in the field of microbiology was Louis Pasteur, 1822 to 1895, and he's also called as the father of microbiology. Here you can see that he's working on the technology of fermentation. Next, he has here in this image, he's working on the rabies vaccination. So, as I have already discussed, that he made a groundbreaking contributions in the field of microbiology, and is also called in mark of respect. He is also called as the father of microbiology. So, what was the germ theory of diseases? So, Pasteur work was foundational to the germ theory. The idea that specific microorganisms cause a specific disease means that. Suppose, for example, if we say that staphylococcus are responsible for causing skin diseases, whereas the E. coli bacteria are mostly responsible for the intestinal infections. So here it was, he told that it was the through the germ theory, the idea that that is the diseases, the specific diseases or specific diseases was caused by the specific microorganism. His research demonstrated that diseases were caused by microorganisms invading the body, a revolutionary concept at the time when many believed that disease arose spontaneously from within the body or bad air. So earlier it was believed that usually some bad air, which was also called as the miasma, patients get infected, or within only they develop some diseases. So he was the first to revolutionize this concept that basically the diseases are caused by the microorganism which enters into the body, invading the body. Then coming to the disapproving spontaneous generation, using his famous swan-necked flask, Pasteur conducted definitive experiment that proved that life does not arise spontaneously from non-living matter. He showed that if a sterile broth was not exposed to airborne dust and germs, it remained free of microbial growth, thus establishing the principle that all life comes from pre-existing life. So some of the important contribution of Louis Pasteur was that he developed the process of heating liquids like wine, beer, and milk to a specific temperature for a set of period to kill harmful bacteria and prevent spoilage. The process, now named after him, is widely used today for food safety. Then development of vaccines. Pasteur pioneered the use of weakened microbes, or also called as the attenuated microbes, for vaccination. His key vaccine development included chicken cholera. He discovered that aged bacterial cultures could lose their virulence but still provide immunity against the disease. Anthrax, he believed a successful vaccine. He developed a successful vaccine for anthrax, a deadly disease affecting livestock, through public demonstration at Poly Fort, the place. Here, the main aim of and of Louis Pasteur developing the vaccine was that he believed that if a bacteria is subsequently subcultured, finally after some generation of culture, it will lose its the capacity of causing infections. But it will help in developing the immunity of the body. So it was a very safe method of developing the or boosting the immune mechanism of the body, and this was the foundation stone for any vaccine development. Next important, as already I have discussed about the development of the rabies vaccination, and this was perhaps his most famous achievement in vaccination. He developed the first vaccine for rabies and successfully used it on 9-year-old boy Joseph Mister in 1885, saving his life and ushering in the era of preventive medicine. So these were some of the important contribution in the development of vaccines. Next was the discovery of anaerobiosis. While studying fermentation, he discovered that some microorganisms can live and function only in the absence of oxygen, a phenomenon known as the Pasteur effect. He introduced the terms aerobic and anaerobic to classify organisms based on their oxygen requirements. Next important development was aseptic techniques and sterilization. His work led to the introduction and promotion of sterilization techniques and the use of aseptic procedures in medical practices, which significantly reduced post-operative infections and mortality rates. Whatever we see the different process of sterilization, or basically the technique here, he wanted to say that any object or any instrument which we are using in the medical practices should be free from microbes. So as and how we can free it, and what are the steps needed so that it can be freed from the different type of contagious microbes. And it was a landmark discovery which helped very well in eliminating the post-surgical or post-operative infections, and thus also decreasing the mortality rates among the patients. And in 1887, the Pasteur Institute was founded in Paris to treat rabies victims and serve as a world-renowned center for research in microbiology, which he directed until his death. Next important discoverer was Ignaz Semmelweis in 1818 to 1865. Ignaz Semmelweis' primary contribution to microbiology was the discovery that hand washing could prevent the spread of infections. See, we already know that the term prevention is better than cure. So here we can see that if usually Pasteur only coined that if the diseases which are caused was mostly by the invasion of the microbes inside our body. So if these that the several methods through which we can prevent the spread of these microbes, or non-invasion of these microbes in our body, it can greatly reduce the infections and diseases which are caused by the microbes. So the discovery either made by the Joseph Lister or Pasteur himself, and the next contribution by Ignaz Semmelweis. All these discoveries led to the conclusion that different aseptic techniques greatly help in minimizing the spread of the microorganism. So what was the contribution was Ignaz Semmelweis was that hand washing was important step to prevent the spread of infection, and he demonstrated that puerperal fever or childbed fever was transmitted from physicians performing autopsies to new mothers, and that this could be drastically reduced by requiring doctors to wash their hands with chlorinated lime solution. So here those doctors which were operating or attending the childbearing mother, and earlier they have done some autopsies or some surgical procedures could easily transmit the infection to them. So he proposed that during this procedure, if the doctors use hand washing procedures and using and that also through chlorinated lime solution, the spread of puerperal fever can greatly be reduced. So while he was not a microbiologist, his empirical findings had laid the foundation for antiseptic procedures and germ theory by showing that invisible particles transmitted from corpses causing diseases, a concept that became a cornerstone of modern infection control. Next important discovery which was made by the Koch. Robert Koch was a German scientist, 1843 to 1910. His famous contribution was to the field of medical science or medical microbiology was the given postulates that was called as the Koch postulate. So it was basically divided into four parts. So first postulate was that the microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organism. So if a person is healthy, in those cases, the microorganism which is causing that disease, a particular disease, should not be found, or if it is found, it should not be in abundance. But in a diseased individual or a diseased person, organism, the microorganism which is responsible for causing infection should be present in abundance. Next major postulate was that the microorganism must be isolated from a diseased organism and grown in pure culture. So if it is isolated and then it is grown, if it is inoculated on a culture media, so it can be obtained in a pure culture. Pure culture means that only one type of microorganism should be obtained. Third was that the cultured microorganisms should cause disease when introduced into a healthy organism. So when these bacteria is inoculated, which is obtained in a pure culture form, if inoculated into a healthy individual, can cause the same type of disease which was present in a diseased individual. And fourth postulate was that the microorganism must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent. Now again, when these microorganism, when isolated from the healthy individual who was infected, and again he developed that symptoms, and again from that person or from that organism, if the organism is isolated, it is identical or similar which was isolated in the pure culture form, which was first isolated in the pure culture form. That same microorganism, when inoculated into a healthy individual and when he developed the symptoms, then again that only microorganism could be re-isolated from the diseased person. So this was the important postulate of the Koch. So if we summarize in short, what was the four rules of the Koch postulates? So first was that the microorganism must be identified in all individuals affected by the disease, but not in healthy individuals. The microorganism can be isolated from the diseased individuals and grown in culture. When introduced into a healthy individual, the cultured microorganism should cause disease, and the microorganism must then be re-isolated from the experimental host and found to be identical to the original microorganism. So these were some of the important postulates, but here also there was some limitations in the Koch postulates. And what was some of the important limitations? Here, first was that viruses were not yet to be able to be cultured during the 1800s. So we have already seen that this postulate mostly is present in the case of bacteria, mostly in the bacterial cases, but not in the applicable to the bacteria, but not in the viruses, infections caused by viruses, because still viruses were not able to culture during the 1800s. Second was that the third postulate stipulates that the experimental host should exhibit disease and not must, because it is not necessary that the pure culture which is inoculated into the second host should may or may not show symptoms of those diseases, and it is basically depends upon the host immunity and its resistance power. So it is not necessary that if the the pure culture obtained from the first infected individual could show the same symptoms, though the microorganism may be present, but due to the good immunity of the second host, the it is may or may not be possible that the person may be symptomatic. The individual may be asymptomatic also. Next is that the Koch postulate do not account for prion diseases and other agents that cannot be grown in culture. Another most important exception of this that is especially in the bacteria, that the most of the human bacterial pathogens satisfy Koch postulate except for those of Mycobacterium leprae and Treponema pallidum, which are the causative agent of leprosy and syphilis, and these both bacteria cannot be grown in a artificial culture medium. Okay, artificial means that the chemicals which we use or the different type of extracts we use for preparation of culture medias. So for that we need cell culture medium. We need a live cell to grow them. So these two bacteria do not satisfy, or the Koch postulate is not applicable on Microbacterium leprae and Treponema pallidum. Therefore, the Koch postulates have subsequently been revised to account for recent molecular advances and are no longer an absolute requirement of infectious causality, but to an extent, it helps to understand the mechanism of disease. And when it was postulated, it was a very important part that Koch postulates were in important postulates or important steps to understand the mechanism of the disease. Next, some of the other key contributions of Robert Koch was that he developed some of the important bacteria, especially the anthrax, Bacillus anthracis, where he also discovered the pathogen's dormant spore stage, explaining how the disease could persist in soil. Means that in addition to the hosts, it can also survive outside the host body. Second was Mycobacterium tuberculosis, which was a groundbreaking discovery for which he was awarded the Nobel Prize in Physiology or Medicine in 1905. Next important discovery was Vibrio cholerae, the causative agent of cholera, in 1883. And it was identified during his expedition to Egypt and India, where he linked the spread of the disease to contaminated water sources, leading to public health measures. So all these bacteria once wreaked havoc in the area where they were prominent. So finding, especially see the Vibrio cholerae, when he found that contaminated water sources are major causes of causing the cholera. So it was an important step to identify that and to help in its elimination. Koch postulates that established a systematic four-step methodology, and it was formalized by his assistant Frederick Loeffler to scientifically prove that causal relationship between a specific microorganism and a specific disease. These postulates remain a cornerstone of infectious disease research and are still used as guidelines today. Next important, though we focus mostly on Koch postulates, but he one more foundation or one more theory was laid by the Koch, which is known by the name of Koch phenomenon. So what was the Koch phenomenon? So Koch phenomenon was basically related to the immune response of the body, and that also to the mycobacterium tuberculosis, to a subsequent exposure to the bacterium or its protein extract. And this reaction is characterized by rapid, severe local inflammatory response, often with necrosis that develops quickly and heals relatively fast. So Koch phenomenon basically discusses, or it is mostly it is concerned with the subsequent infection with or subsequent inoculation of mycobacterium tuberculosis, or when a person is subsequently exposed to mycobacterium tuberculous bacterium or its protein extract, and what are the reaction which is seen in the body, which is mostly seen as a local inflammatory response, and that develops quickly, and also it heals relatively fast. So discovery and description. Robert Koch observed this phenomenon during his tuberculosis research in the late 19th century. He noted that following differences in guinea pigs. A healthy guinea pig inoculated with tubercle bacilli would develop a slow growing lesion that eventually led to widespread disease and death. A guinea pig that had been previously infected with tuberculosis, when re-inoculated with the same bacilli or a glycerin extract of the bacteria, which he called tuberculin, developed a distinct accelerated reaction at the injection site. The area would quickly become dark, necrotic, and form an ulcer, but then it would heal rapidly, and the animal would not develop a new systemic infection from the second injection. You can say that it was also a type of helping or boosting the immunity in an organism which was already infected. And these observations led to Koch to hypothesize that the initial infection conferred a degree of immunity that modified the response to subsequent exposure, which he termed acquired immunity, and therefore it was also a landmark in the discovery of the field of sub part of microbiology, which is also an important part to study the immune mechanism within the body and how it develops to the certain infections. So it was the major contribution of Robert Koch. So it is also, the Koch phenomenon is also a classic example of a type 4 delayed type hypersensitivity reaction mediated by sensitized T cell. The reaction is a strong localized immune response to the mycobacterial antigens. While Koch initially hoped tuberculin would be a cure for tuberculosis, it proved ineffective for therapy and could even be harmful in patients with advanced disease due to the severe tissue damaging inflammatory response it caused in sensitized individuals. So every experiment have its own limitations, and it was seen that in patients which have already progressed to an advanced stage of the disease, there it can cause a strong inflammatory responses and even damage the body cells and tissues. However, his discovery led to the development of the tuberculin skin test, which is also known by the name of MON2 test, where a small amount of purified protein derivative PPD from tuberculin is injected into the skin to test for preventive exposure to Mycobacterium tuberculosis or BCG vaccination. So it was a very milestone in the development of BCG vaccination, and a positive reaction in duration or swelling indicates a present or past immune response. Now, how it is distinction from Koch postulates? The Koch phenomenon is distinct from Koch postulates by a set of criteria established by Robert Koch to determine if a specific microorganism is the cause of a particular disease. The postulates include requirement that the microorganism be found in diseased individuals, but not healthy ones, be isolatable and grown in pure culture, cause the same diseases when introduced to a healthy host, and be re-isolatable from the host. While Koch postulates remain fundamental in microbiology, the Koch phenomenon specifically describes an immune response to the tubercle bacillus. Major contributions of Robert Koch is the development of discovery of anthrax diseases, tuberculosis, and cholera bacilli. Koch observed the phenomenon of acquired immunity through the, as already we discussed, that Koch phenomenon. And other major contribution was that he introduced solid media for culture of bacteria. Koch pioneered the use of agar as a base of culture media. He developed the pour plate method and was the first to use solid culture media for culture of bacteria. Koch also developed media suitable for growing bacteria isolated from the body. Because of their similarity to the body fluids, meat extracts and protein digests were used as nutrient sources. The result was the development of nutrient broth and nutrient agar media that are still in wide use today. He also introduced methods for isolation of bacteria in pure culture. He described hanging drop method for testing motility. He introduced staining techniques by using aniline dye. He invented the hot air oven and steam sterilizer, also, and also introduced methods to find out efficacy of antiseptics. So, especially in the field of bacteria, the development made by the Robert Koch is commendable, and all those methods we discussed now are still in use in the field of microbiology. So Robert Koch is also called as the father of bacteriology. Louis Pasteur is known as the father of microbiology, and Robert Koch is known as the father of bacteriology due to his commendable contribution in the in developing different methods for the isolation of some of the important bacteria. Next, other important contributions were made by Emily Rocks 1853 to 1933 and Alexandre Yersin 1863 to 1943. Both were the French bacteriologists. And in 1888, Rocks joined the newly created Pasteur Institute, where with Alexandre Yersin, he demonstrated that the symptoms of diphtheria are caused by a toxin secreted by the diphtheria bacilli. So here new concept came, and that was the concept of toxins. Toxins are basically the poisonous substances which are secreted by some of the bacteria. And on this, the contributions were, the work was done by the scientist Emily Rocks and Alexandre Yersin. And the work was done on the diphtheria bacilli, which secretes the diphtheria toxins. And the discovery, together with the subsequent finding by bacteriologist Emil von Behring and Kitasato Shibasaburo, that infection with the diphtheria bacillus elicits the production of an antitoxin, led to the development of diphtheria immunization and serum therapy. Rocks became director of the Pasteur Institute in 1904, a post he held until his death in 1933. Now coming to the era of 1900s, from 1918 to 1919, a Spanish flu pandemic killed an estimated 20 to 50 million people worldwide, including 1 in 67 United States soldiers, making an influenza vaccine a US military priority. Early experiments with influenza vaccines are carried out. The US Army Medical School tests 2 million doses in 1918, but results are inconclusive. See, as we say that the 19th century was the era of development in the era of bacteriology, whereas the 20th century can be best regarded as an era of development of virology, which was also a landmark in the diagnosis or development of different viral techniques for isolation and treatment of different viral diseases. So first development was made by 1937. Max Tiller, Hugh Smith, and Eugen Hagen developed the 17D vaccine against yellow fever. And the vaccine is approved in 1938, and over a million people have received in that year. Tiller goes to be awarded the Nobel Prize. In 1939, bacteriologist Pearl Kendrick and Grace Eldring demonstrated the efficacy of the pertussis whooping cough vaccine, and it was the bacterial vaccine. Whooping cough the cause of the whooping cough was Bordetella pertussis, a bacteria. The scientists showed that vaccination reduces the rates at which children get sick from 15.1 per 100 children to 2.3 per 100. Next, in 1952 to 1955, the first effective polio vaccine is developed by Jonas Salk, and trials begin. Salk tests the vaccine on himself and his family the following year, and mass trials involving 1.3 million children takes place in 1954. By 1960, a second type of polio vaccine developed by Albert Sabin is approved for use. Sabin's vaccine was live attenuated using the virus in weakened form and could be given orally as drops or on a sugar cube. The oral polio vaccine was first tested and produced in the Soviet Union and Eastern Europe. Czechoslovakia becomes the first country in the world to eliminate polio. In 1969, 4 years after Dr. Baruch Blumberg discovers the hepatitis B virus, he works with microbiologist Irvin Millman to develop the first hepatitis B vaccine using a heat-treated form of the virus. A plasma-derived inactivated vaccine is approved for commercial use from 1981 to 1990, and is genetically engineered vaccine developed in 1986 is still in use today. In 1971, the measles vaccine is combined with, which was discovered in 1963, is combined with recently developed vaccine against mumps in 1967, was discovered the mumps vaccine and rubella in 1969 into a single vaccination MMR by Dr. Morris Hilleman. In 1978, a polysaccharide vaccine that protects against 14 different strains of pneumococcal pneumonia is licensed. In 1983, it is expanded to protect against 23 strains of pneumonia. In 1985, the first vaccine against disease caused by Haemophilus influenzae type B is licensed after David H. Smith founds a company to produce it. Smith and Porter W. Anderson Jr. had been working together on vaccination since 1968. So these are some of the important contributions in the field of microbiology as well as in the field of vaccine development and treatment. I hope this had certainly helped you for information on the development historical part of the microbiology field. Thank you.